Okay, so we've got a new pen. Very exciting. So today we're going to talk about endothelial cells. Endometrium. We're going to talk about the endometrium and all of the stem cells in there. We're going to get a little bit of anatomy, and then we'll focus on the parts of the anatomy that are relevant to the stem cells in the endometrium, because there are 3, maybe 4 different kinds of stem cells in there. Then we'll talk about the clinical applications and the diseases of the endometrium. So let's get into it. So obviously the endometrium needs to regenerate a lot, right? So it regenerates, 400 to 500 times in a woman's lifetime. And that's important because we need something that's really robust as we regenerate. We need a regeneration approach that not only can produce a whole lot of tissue every month in a 1 week period, but we need to do it robustly. So how is it regulated? So estrogen causes proliferation. Progesterone causes secretion. And in the absence of a pregnancy, the absence of these two, well, I guess that's the wrong way, but the absence of these two causes the shedding of the endometrial functionalis layer. Cool. So now we'll talk a little bit about the layers of the endometrium. That's a good transition. What is the functionalis layer? Right. We'll look at that anatomy very quickly. So your endometrium has 2 main layers inside of the mesometrium, I think it is. So let's call this is the basal layer, and this is the functional layer, otherwise known as functionalis and base basalis layers. Making sense. Okay, and so then we can draw within this lovely little diagram. Yeah, okay. Chronogenic experience. A couple of other interesting things about the endometrium. So we looked at the markers that make up these the stem cells therein, and it's pretty standard. But OCT4, SOX2, OCT4, SSEA4, and what else have I got? NANO and CKIT. Pretty standard cell markers for stem cells. But those are cell markers for endothelial stem cells. So we were talking about these layers, and these layers have these cysty things, glands. They have glands, and the glands have stem cells in them. But in addition to the glands, and this is important, we have veins running through the functionalis layer. And I'm using all my colours, which is very exciting. And around those veins, we have cells. We have cells of various kinds. There are actually 2 different kinds of cells around those veins, which we're going to talk about in a minute. Okay, so in this diagram, there are 3 different kinds of stem cells, and this is really important, okay? So in the endometrium, there are 3 different kinds of stem cells which we're going to talk about, okay? And that's quite unusual, right? Because all the tissues that we've talked about so far have 1 kind of stem cell if you're lucky, and then you need to coax them out. But this tissue has 3 different kinds of stem cells. So that's really interesting. And then kind of maybe a 4th kind that we'll get to in a second. But this is really cool. Okay, so what are the 3 kinds of stem cells, Ryan, you ask? And rightly so. And so the three kinds of stem cells are epithelial endometrial stem cells. I think I got that right. Yes. Yeah, endometrial epithelial stem cells. So endometrial epithelial stem cells, right? Endometrial epithelial stem cells, which is cool. We have endometrial mesenchymal stem cells, which are these boys or girls. Endothelial mesenchymal. And then finally, and I don't have a different color, but I'm going to do it in red. We have side population cells. Right? Okay, so we have three different kinds of cells that side population cells kind of, if you drew a Venn diagram, right, of stem cells or side population cells and stem cells, the Venn diagram would kind of look like that. Like not all side pop cells are stem cells and not all stem cells are obviously side pop cells, but side pop cells and stem cells have a reasonable amount of overlap. Just so you know. Okay, so those are the three kinds of stem cells that we have here. And we're going to go through them. We're going to go through them and talk about each of them. Starting with the endometrial mesenchymal stem cell. So the endometrial mesenchymal stem cell is actually pretty much a mesenchymal stem cell. It just happens to be in the endometrium, which is cool. So it's an MSC in the endometrium. And we've done a lecture about mesenchymal stem cells. So they give rise to the skin, the bone and fat. And in this case, they just happen to exist here and provide stromal support. And they seem to have quite a strong vasculation role. Right? They have a vasculation role, which is pretty cool. Which is, I would say, useful. I'm going to pause here right there. Okay, and we have a lecture about mesenchymal stem cells, so you can look that up if you want to learn more about mesenchymal stem cells. So we'll talk about the endometrial epithelial progenitor cells. They're not stem cells. They're progenitor cells, but they are the earliest in the epithelial progeny, or the endometrial growth line. Okay, so they give rise to all of the cells in this gland as they mature through their entire lineage, but these are the basic ones that live at the bottom of the gland, here, hanging out, doing the thing, giving rise to all the cells that come up here. Okay, and that's really straightforward. They can produce kind of spheroids when they're cultured in 3D, and that's about all we need on those. And finally, there's the there's the side population cells. Side population cells are interesting. So side population cells are cells enabled with a particular protein, a particular enzyme that allows them to pump a particular kind of dye and other materials out of the cell. So there's a dye that we use and we put it into the medium, and other cells will just kind of reach a osmotic static point with the dye around them, and these cells in fact will pump it out. And like I said, there's a reasonably kind of large overlap between stem cells and these cells, but not all of these cells are stem cells, and not all stem cells are these side population cells. An interesting thing to consider with side population cells is that when we have cancer cells, a lot of these cancer cells are likely to have side population properties, which means that if you have cancer stem cells which have side population properties, they, instead of taking up your chemotherapy drug and dying in the way that they were supposed to, they don't. They just pump it out. And so you can't kill the cancer stem cell, which is the most dangerous of the cancer cells. So that's really suboptimal and an interesting challenge. The other thing to note about the endometrial epithelial progenitors, these guys, is that they don't have any receptors for female hormones. They don't have any receptors for progesterone or estrogen, which makes sense because they're the earliest in that lineage and they don't need to interact with that system. Their later, more progressed cells will interact with those hormones and excrete the functional layer and progenerate into the functional layer, or they will receive those signals. So these guys down here really don't interact with that system at all. They just kind of grow. And they interact with N-cadherin, so you can pull them out with N-cadherin. Subpopulation cells, yep. Okay. There's an antibody for side population cells, if any of you care. It's ABCG2. ABCG2. And that, I think, is a decent summary of that particular piece of anatomy. Okay, now, interesting. There's that fourth kind of stem cell that I mentioned, which I'm going to do over here. So the fourth kind of stem cell that I mentioned was bone marrow. What? Wild. Right, so bone marrow. So it's been shown that if a woman gets an injection of bone marrow, it's possible to develop chimeric populations of these endometrial epithelial progenitors, in their tissue, which is really interesting because what it means is you're getting stem cells coming from the bone marrow, migrating into this tissue, and then grafting on. That's a bone. And then grafting on, and then creating these chimeric populations in the glands. It also means that there are multiple stem cells in these glands, which give rise to the chimeric populations above. But it's really interesting and kind of unique that there's this grafting of a totally different tissue from bone marrow. So just a note about those endometrial epithelial stem cells. Is that they're quite easy to de-differentiate into induced pluripotent stem cells. It can be done a couple of weeks more quickly than if we took progenitors out of the skin, or stem cells out of the skin, which is a nice little shortcut. If you have this kind of tissue accessible, it's nice to be able to make those stem cells a little bit more quickly, and then they can be differentiated into whatever you want. Now, therapeutic uses. Therapy. So, what do we do with these stem cells for therapy? Well, they have a bunch of interesting properties, okay? These endometrial kind of stem cells and progenitors have a lot of kind of interesting properties that allow them to be quite useful for therapeutic purposes. So they kind of have some immunoprivilege, which is important, and we think that they have immune, and they also have a low immune response. So they have a low immune response because they're having to interact with a fetus which has foreign antibodies from the father, which makes sense. And they have some immunoprivilege, which is just handy because it means that we can kind of inject them and get less of an immune response than we might expect. So we can use them as immunoregulatory agents. Use them for immunoregulatory agents. So they reduce inflammation in cystic fibrosis. Okay? They release cytokines. Do they release or inhibit the release of... The kind of inflammation that they reduce is neuroinflammation. That's important. They can release cytokines which reduce... They can reduce pro-inflammation cytokines. So they reduce fewer cytokines. Reduce pro-inflammation cytokines. Right? Reduce pro-inflammation cytokines, and they reduce infiltration of macrophage. Reduce infiltration of macrophages. Cool. So in all of that way, they kind of do their immunoregulatory approach. They use these cells as immunoregulatory devices, things, agents, and that's useful in a range of cases as we've seen. So, okay, and this is the last bit of the therapeutic applications of these cells. But we can use them to make hepatocytes, which obviously are useful in liver tissue. So we can't use them to make liver stem cell progenitors or liver cell progenitors or what was the other one? I forget. Doesn't matter. But that's useful. We can inject them in IV. We can inject them in IV, which will help us fight hypoglycemia in type 1 diabetes in mice. We can use them to fight premature ovarian failure. And we can also use them to reduce the stroke phenotype. So if they get into your brain, they can increase the functionality of stroke neurons, which is cool. And now we'll just talk about one last condition, which kind of is interesting and relevant and just a little tidbit. So, endometriosis. Where are we going to do this? Blue, I think. Endometriosis. Let's try and draw a uterus. It'll be terrible, I can assure you. Alright, so get your fallopian tubes and away you go. Yeah, whatever. And so you have this tissue. You have the endometrial tissue, right? And every month it gets evacuated, and that's cool. And this tissue, you really want it to stay here. You really want it to stay here in the uterus and not go in a retrograde direction and engraft. But that's exactly what endometriosis is with a little bit of retrograde flow, you start to get these bodies growing back here, which is bad and uncomfortable and just a whole bunch of negative things. But it's interesting because those women with more, because all women seem to get retrograde flow, but those with more STEMI cells, get endometriosis. So why is that? Well, it's because as these cells move up, and like all of them move up, these ones, which are more STEMI, tend to engraft more effectively and cause the endometriosis, which is pretty interesting. Yep, and that's our whole lecture. That's our whole lecture on the endometrium. Thanks, guys.